You know that feeling. You've got this stack of articles, maybe some research papers, notes. Could be this something someone sent you or, stuff you found online. And it's all about this one topic that seems really interesting.
Yeah, like it's got potential, maybe some breakthroughs or surprising facts hidden in there.
Exactly. But then sorting through it all, trying to figure out what's really important, what's actually backed up by good evidence and what does it all mean, that can feel like a whole other job.
It's definitely the modern challenge, isn't it? There's just so much information flying around, but getting that distilled, actionable knowledge, that's much tougher. You have the pieces, but seeing the whole picture, understanding the nuances, that takes some real digging.
Right. It requires a deep dive. And that's exactly what we do here. We take your stack of sources, the specific ones you shared with us, and we jump right in.
We try to navigate the data, pull out the key findings, add some context, connect the dots, basically help you get up to speed, quickly but thoroughly.
The goal is for you to feel well-informed, grounded in the actual sources in a way that hopefully, sticks.
Think of us as your guides for this specific material, helping you zero in on what really matters in the documents we're looking at today.
So today we're diving into something pretty intriguing. Zeolite Clinoptilolite. That's a natural mineral. Might sound a bit technical to some.
Well, maybe not a household name.
Right. But it's actually been around for ages. It's used in all sorts of things, from like environmental cleanup to animal feed and health.
Industrially, it's got a long history. But the picture of what it does inside the human body, that's been a bit fuzzier, especially when it comes to rigorous human data on how it affects things like minerals and metals.
That's the gap, isn't it? There's been traditional use, some promising animal studies for sure, but getting that really solid, controlled clinical data in humans, that's been less available, at least until more recently.
And that's precisely why the sources you've provided are so valuable. We're not just talking generally about zeolites today.
No, we've got specifics. Information drawn from 3 different human clinical trials looking at a particular certified type of clinoptilolite. Plus, some pretty comprehensive reviews that dig into its basic properties, what's been seen in various studies, and even explore some really compelling potential links to complex conditions, things like neurodegenerative diseases.
Exactly. These aren't just opinion pieces. They offer specific data, methodologies, results, conclusions from actual research. It gives us something solid to ground our understanding in.
So our goal for this deep dive is to really unpack these specific sources. Go beyond the headlines. We want to distill the most important findings about this material's effects in humans, based only on these studies and reviews presented.
And explore those surprising connections the reviews talk about, especially around heavy metals, gut health, and maybe even brain health.
Okay, let's get into it. Let's unpack this.
Right.
So where do we even start? The basics, I guess.
Yep.
What exactly is zeolite clinoptilolite, based on what these sources are telling us? Sources 1, 2, and 3 seem to lay this out.
Okay, yeah. At its heart, clinoptilolite is described as a natural inorganic crystal. It's a mineral, and it belongs to a bigger family called zeolites.
And its structure. It seems really important for how it works.
Right.
They call it an aluminosilicate framework. Sounds complicated.
It does sound technical, but the concept is kind of cool. Imagine a very rigid, three-dimensional structure, like a microscopic cage or honeycomb. It's built from silicon, aluminum, and oxygen atoms all linked together.
A 3D cage.
Right. And this structure creates these tiny, stable channels and cavities inside the crystal. Within those little pockets, the mineral naturally holds onto 2 things: water molecules and positively charged ions. Chemists call them cations.
Cations like what?
Things like calcium, sodium, potassium, common elements. They're inside the structure, basically balancing out the overall negative charge of that silicon aluminum framework.
Oh, okay. And it's those trap cations that give it its most well-known property, that ion exchange thing.
Precisely. This is the critical feature, the reason it's interesting for potential health uses. Those cations inside, like the calcium or sodium, they aren't glued in place permanently. They can actually be swapped out.
Looks for what?
For other cations that are floating around in the environment, the zeolite finds itself in, like, say, the fluids in your gut.
And this is where the whole detoxification idea comes from. It swaps good ions for bad ones.
Essentially, yes. Different cations have different levels of stickiness or affinity for the zeolite structure, and it turns out clinoptilolite has a particularly strong attraction, a high affinity, for certain potentially harmful cations.
Like heavy metals.
Exactly. Things like lead, Pb, cadmium, Cd, nickel, Ni, even arsenic, as are mentioned. So when clinoptilolite encounters these metals in its environment, it tends to grab onto them more tightly than its own internal cations.
So it does a trade. It releases, say, calcium and grabs on a lead instead.
That's the basic mechanism described in the sources. It swaps its own, less tightly bound cations for the heavy metal ions, effectively trapping those contaminants inside its cage-like structure while releasing potentially less harmful or even beneficial ions like calcium.
Okay, so it acts like a kind of molecular trap, selectively binding these unwanted, positively charged things. Where does this stuff come from naturally?
The sources mention it's typically volcanic in origin. It forms over, long geological periods when volcanic ash or glass reacts with alkaline groundwater.
And it's stable, like it doesn't just break down in the stomach.
That's a key point mentioned for its use inside the body. It has high thermal stability, and it's resistant to chemical breakdown, particularly in acidic environments like the stomach, so it's expected to pass through the digestive system largely intact.
Got it. Stable, natural mineral, cage structure, ion exchange power, grabs heavy metals. Now, the sources also make a point of distinguishing between just natural clinoptilolite and specifically modified forms, right?
They do. This is important. Sources 1, 3, and 4 talk specifically about something called PMA-Zeolite. PMA stands for Panaceo Microactivation.
So this isn't just like crushed volcanic rock.
No, it's described as a registered and certified material that goes through a specific manufacturing process. That process is called tribomechanical activation.
Tribomechanical, what does that involve? Sounds intense.
It's basically a very high-energy milling or grinding process. Source 4 describes it as patented. Think of it as subjecting the mineral particles to intense mechanical stress and friction.
And why do that? What's the point of this activation?
The idea behind it, as described, is to potentially enhance the material's biophysical properties, maybe increase its effective surface area or change how accessible those internal channels are, theoretically making it work better, making it more reactive or efficient in its ion exchange or other interactions.
Okay, so optimizing it. But Source 1 had a really important warning or limitation about this, right? About generalizing the findings.
Yes, absolutely crucial point. Because this specific activation process might significantly alter the material's properties compared to just raw clinoptilolite or clinoptilolite processed in a different way.
You can't just assume.
Exactly. You can't just assume that the results they found using this specific PMA zeolite material will automatically apply to any other zeolite or clinoptilolite product out there. You'd need separate studies on those other materials.
Okay, that's crystal clear. The research we're digging into today is specifically about this tested PMA zeolite. So why the big push for these specific human studies mentioned in Source 1 and 2? We knew it was used in animals industry.
Well, as Source 2 points out, it has been studied quite a bit and used in, say, veterinary medicine and agriculture for a long time. There's documented evidence in animals suggesting positive effects, things like antioxidant support, immune stimulation, helping with diarrhea, maybe bone health, even some anti-tumor effects mentioned in reviews.
So lots of hints from animal studies that it's beneficial and probably safe for them. But humans are different.
Humans are different, and the standards for evidence are higher. Source 1 really highlights the lack of comprehensive, controlled clinical data in humans looking at the systemic effects, like what actually happens throughout the body after you swallow it.
And they were particularly interested in what? Minerals and metals.
Yes, especially how it affected the levels of essential physiologically important minerals, iron, calcium, zinc, etc., in the bloodstream. And also, critically, the levels of potentially harmful contaminants like heavy metals.
And this was driven by a specific worry or hypothesis, wasn't it? Related to that ion exchange thing.
Exactly. It's laid out in Source 1 as a key rationale. If this stuff is so good at swapping ions, could it potentially grab onto and deplete the body's essential minerals along with the bad stuff? That was one concern.
Okay, could it steal our good minerals?
Right. And the flip side was, could contaminants within the zeolite material itself, because it's a natural mineral, it might contain traces of things like aluminum or lead, could those leak out of the zeolite and into the bloodstream after you ingest it?
Okay. So, two main safety questions. Does it take away good stuff, and does it release bad stuff?
Those were the core working hypotheses driving the need for these rigorous clinical trials in humans to monitor these possibilities carefully.
Which brings us neatly to the overview of the 3 clinical trials that Source 1 describes, all designed to look at the effects of this specific PMA zeolite.
Right. So the first one they detail is the MMBP study. MMBP stands for Mineral Metabolism and Selected Blood Parameters.
Okay, MMBP, what was the setup?
This was a short-term study, just 28 days. It was in healthy volunteers and was mainly an exploratory safety study. They used a dose of 6 grams per day. And interestingly, they included 2 groups, people trying it for the first time, the naive group, and people who had already been using it long-term, the chronic group.
Okay, short-term, healthy people, first-timers versus long-term users. What was next?
Next was the Morbus Crohn study. Morbus Crohn is another name for Crohn's disease. This was a medium-term trial running for 12 weeks.
Longer duration. And who's in this one?
It included patients who had uncontrolled Crohn's disease and also a group of healthy volunteers for comparison. This study was more rigorously designed. Randomized, placebo-controlled, and double-blind. They used the same dose, 6 grams per day. Source 1 calls it a pilot study, so probably smaller numbers of participants.
Right, standard design for testing effectiveness. And the third one?
The third was the osteoporosis TOP-P study. TOP-P stands for Treatment of Osteoporosis by Panaceo. This was the big one, the longest trial. It ran for up to 4 years.
4 years, wow. And this was in?
Patients diagnosed with osteoporosis, like the Crohn's study, it was randomized, placebo-controlled, double-blind. But notably, they used a higher dose in this trial, 9 grams per day.
Higher dose, longer time, specific patient group focused on bone health. But they still measured minerals and metals.
Oh, yes. While the main focus might have been bone-related outcomes, Source 1 makes it clear they did extensive monitoring of blood parameters, including minerals and metals using standard lab tests, and also more sensitive methods like ICP-MS across all 3 studies. They list the parameters in Table 1 and the reference values they used in Table 2.
Okay, so 3 distinct studies, different populations, different durations, slightly different doses, but all looking closely at blood chemistry, minerals, and metals with this specific PMA-Zeolite. Let's dig into the findings, starting with that first one. The MMBP, Study Healthy Folks, 28 days. What were the key results on minerals and contaminants?
This study really helped tackle those primary safety questions. First, looking at the essential minerals, iron, sodium, potassium, calcium, magnesium, zinc, measured with standard tests. In the people who were already long-term users, the chronic group, they found no clinically relevant changes over the 28 days.
Meaning their levels stayed pretty stable.
Yeah, their levels basically stayed within the normal reference range. So for those already using it, it didn't seem to be throwing their basic mineral balance out of whack in the short term.
Okay, that's reassuring for long-term users.
Yeah.
What about the newbies, the naive group?
For the new users, there was actually a kind of interesting observation. Source 1 notes that some individuals in this group started the study with certain blood values slightly outside the normal range, maybe low iron or slightly high zinc or liver enzymes, things like that.
Okay.
And for some of these people, after taking the PMA-Zeolite for 28 days, those initially altered values actually showed improvement or normalized. They moved back towards or into the normal range.
Huh. So not depletion, but maybe regulation in some cases.
Right.
That's a potential upside, even in a safety study. Were there any statistically significant differences between the naive and chronic groups for minerals?
Yes, they did report some statistically significant group differences for calcium, magnesium, and sodium at certain time points. But, and this is the really critical part for that depletion concern, despite these statistical differences between the groups, the actual measured levels of these minerals in both groups stayed within the normal clinical reference range for the whole study.
So the math showed a difference between groups, but nobody's levels actually dropped into a dangerous or deficient zone.
Exactly. The researchers concluded these differences weren't clinically relevant. They didn't indicate a health risk in these healthy volunteers over this time frame. It was a key finding arguing against the idea that it depletes essential minerals.
Okay, that addresses one major safety question. What about the other one? The idea that contaminants from the zeolite might leak into the blood.
The evidence from this MMBP study did not support that concern over the 28 days. In fact, they saw the opposite. For some metals, they saw signs of detoxification.
Detoxification, like what?
Levels of mercury, Hg, and cadmium, Cd, 2 well-known toxic heavy metals, actually decreased significantly in the naive group over the 28 days of taking the PMA-Zeolite.
Wow, decreased. That's definitely not leaking out. It sounds like it's pulling them in.
That's the implication. The authors noted that baseline cadmium levels seemed linked to smoking habits in some participants, and mercury arsenic might have been related to diet, like fish consumption. But whatever the source, taking the supplement was associated with a statistically significant reduction in mercury and cadmium in the blood of these new users.
Okay, so mercury and cadmium went down. What about other metals like lead or aluminum in this short MMBP study?
Lead PB levels did show a slight increase in the naive group after 28 days, but crucially, the levels remained well within the normal reference range. Aluminum Al and nickel Ni levels were generally low, below or within reference values.
A slight increase in lead, but still normal. But what about that immediate leakage idea? Did they test for that?
Yes, and this was a really important safety check detailed in Source 1's discussion. They specifically tested blood levels of aluminum and lead just 1 hour after the very first dose in the naive group.
And
Any sudden spike?
Nope. No increased levels of either aluminum or lead were detected just 1 hour after taking it. This strongly suggests that these metals weren't immediately leaching out of the material and getting absorbed into the bloodstream upon ingestion.
that's a critical piece of safety data. So to sum up MMBP, 28 days, healthy people, showed basic safety, didn't seem to deplete essential minerals into concerning ranges, didn't show immediate leaching of contaminants like Al or PEB, and actually showed decreases in mercury and cadmium.
That's a very good summary of the key takeaways from that first study. It provided initial reassurance on the main safety hypotheses.
Okay, let's move on to the Morbus Crohn trial. This was longer, 12 weeks, and included patients with active Crohn's disease as well as healthy folks. What did this one add to the picture regarding safety and metal mineral status?
Much like the MMBP study, this trial found that standard blood parameters and the levels of most minerals and metals measured were not significantly affected in any group, not in the Crohn's patients taking zeolite, not in the Crohn's patients taking placebo, and not in the healthy volunteers over the 12-week duration.
So again, reinforcing the general safety profile, even over a medium term and in a population with inflammatory bowel disease.
Correct. It extended those safety observations to 12 weeks and included a patient group known to sometimes have issues with nutrient absorption or inflammation.
But was there a specific finding related to detoxification in this study, like the mercury cadmium and MMBP?
Yes, there was one key detoxification finding here: arsenic AS levels. In the Crohn's patients who received the PMA-ZC, their blood arsenic levels diminished significantly compared to the Crohn's patients who received the placebo.
Arsenic went down specifically in the supplemented Crohn's patients. Interesting. Did the authors have any thoughts on why arsenic showed up here?
They mentioned diet in the MMBP study potentially influencing arsenic levels. They did comment on that again. They noted that the participants in this Crohn's study were mainly on a continental European diet, likely with lower fish intake compared to the Mediterranean diet group in the MMBP study. This might explain potential differences in baseline arsenic levels.
Well, maybe lower starting levels overall, but the zeolite still made a difference?
Apparently so. Even if baseline levels were different, the supplementation period was associated with a statistically significant reduction in arsenic, specifically in the group receiving the active PMA-ZC treatment compared to placebo.
Okay. So the Crohn's study, safe over 12 weeks in patients and healthy controls, and adds arsenic detoxification to the lift of effects observed in these trials from Source 1.
Pretty much. General safety confirmed for the medium term, plus a specific effect on arsenic removal in the patient group.
All right, now for the longest, highest dose study, the osteoporosis TOP study. Up to 4 years, 9 grams a day, in patients with osteoporosis. How did things look over this much longer time frame? Let's start with safety and standard blood tests.
The good news on the general safety front continued. Source 1 reports that standard blood measurements remained within the normal reference range throughout the entire study duration, up to 4 years, in the patients receiving PMA-ZC.
Okay, so basic liver function, kidney function, blood counts, etc., stayed okay even with long-term higher dose use. What about the essential minerals? In the shorter studies, they stayed mostly within the normal range. Was that still true after 4 years?
This is where the TOP study revealed more complexity. While most mineral levels generally stayed within the reference range, the authors did report statistically relevant fluctuations over the course of the treatment. And more importantly, they observed that the average concentrations of copper, C, sodium, Nef, and calcium, AT, actually diminished to levels below the lower limit of the reference values at certain points during the multi-year study.
Below the normal range. Okay. That sounds potentially more significant than the fluctuation seen before, especially calcium in people with osteoporosis. When did this happen?
It wasn't immediate. Source 1, specifically Figure 2 in Table 5, shows copper levels dipped below range in year 3, but actually came back up into the normal range by year 4. Sodium levels, however, were below the reference range in years 2, 3, and 4. And calcium levels were below the reference range in years 3 and 4.
Sodium and calcium staying below normal for the later years. How did the researchers interpret this? Did they think the zeolite was suddenly depleting these minerals after long-term use?
They proposed a different hypothesis, one that's tied specifically to the context of treating osteoporosis. Their theory, presented in the discussion section of Source 1, is that these decreases in sodium and calcium, and maybe the temporary dip in copper, might actually be related to the bone remodeling process itself.
How so?
They suggest that if the treatment, which included the PMA zeolite, was successfully stimulating bone formation or reducing bone breakdown, then minerals like calcium and sodium, which are major components of bone, might be getting actively incorporated into the bone structure.
Ah. So instead of the zeolite pulling these minerals out of the body and causing them to be lost, the idea is that the body might be pulling them from the bloodstream into the bones as the bones get stronger.
That's the proposed explanation offered in Source 1. It links the observed decrease in blood levels of these minerals to a potentially positive biological effect happening in the bones of these osteoporosis patients. It's an interpretation based on the clinical context.
That's a really interesting alternative explanation. And based on this possibility, did they make any clinical recommendations for people using this long term?
Yes. Based on these findings, the authors included a practical suggestion in their conclusion. They recommend that for patients with osteoporosis who are taking PMA-ZC long-term, specifically defined as more than 1 year, it might be a good idea for their doctors to periodically check their blood levels of copper, calcium, and sodium.
Just to keep an eye on things.
Exactly. And potentially consider adjuvant mineral supplementation if those levels start to trend too low, just to ensure everything stays balanced while the bones are hopefully benefiting. It's presented as a sensible precaution based on their long-term data in this specific population.
That makes perfect sense. Monitor in the context of the disease and treatment. Okay. Now, what about the contaminants lead, aluminum, etc. in this 4-year study? Did they see metals building up over time with the higher dose?
No accumulation was seen. The mean concentrations for the measured metals were consistently within the normal reference ranges throughout the 4 years. However, there was a really interesting finding regarding lead.
Lead, again.
Yeah.
What did they find over 4 years?
They found that blood lead levels were statistically significantly higher in the group taking PMA zeolite compared to the placebo group for the first 3 years of the study, TOP1, TOP3.
Higher. But you said they stayed within the normal range?
Correct. Statistically higher than placebo, but the average levels were still within the accepted normal clinical range. But here's the fascinating twist. In the 4th year of the study, TOP4, lead levels in the PMA zeolite group decreased significantly compared to where they were in the 3rd year.
Wait, so lead was higher than placebo for 3 years, though still normal, and then it dropped significantly in the 4th year. That sounds counterintuitive. If the material was leaking lead, how did the authors explain that pattern?
This finding really fueled their alternative hypothesis about detoxification rather than contamination. They proposed that the initial increase in blood lead observed might actually represent lead being mobilized, essentially pulled out of long-term storage sites in the body.
Like where? Where does lead get stored?
Bones are a major storage site for lead accumulated over a lifetime. The authors suggest that perhaps the bone remodeling process, potentially stimulated by the treatment, was releasing some of this old, stored lead back into the bloodstream.
Okay, so the higher blood level wasn't new lead coming from the zeolite, but old lead coming out of the bones.
That's the hypothesis. And then as the treatment continued over several years, the PMA zeolite could bind to this mobilized lead in the gut, as it's excreted via bile, for example, and facilitate its elimination from the body. This would explain why the lead levels then decreased significantly in the 4th year.
So it's potentially a 2-step process. Mobilize stored lead first, then eliminate it. That paints the initial rise in a very different light as a sign of detox starting, not contamination.
Precisely. That's the interpretation offered in Source 1's discussion and conclusion. It suggests a potential long-term benefit in reducing the total body burden of stored lead, even if blood levels temporarily rise during the mobilization phase.
That's a really important nuance from the long-term data. Did they see any other evidence of detoxification in the TOP study?
Yes, they did. Very clear evidence for aluminum and nickel.
What did they find for those?
After 4 years of treatment, the levels of both aluminum, Al, and nickel Ni were statistically significantly lower in the patients taking PMA zeolite compared to the placebo group.
Lower. Okay, that's straightforward detoxification.
Absolutely. This finding provided strong, long-term confirmation of the aluminum binding properties often attributed to Clinoptilolite, and it added nickel detoxification to the list based on this robust, long-term controlled trial.
What about the other metals measured, like arsenic, cadmium, mercury?
In this particular study population, osteoporosis patients, higher dose, long duration, the levels of arsenic, cadmium, and mercury remained within the normal range and didn't show statistically significant differences between the zeolite and placebo groups over the course of the study.
Okay, so the key detox findings in top P were for aluminum, nickel, and that interesting mobilization then reduction pattern for lead.
Correct.
Let's try to synthesize the clinical evidence from all three trials presented in Source 1. Stepping back, what's the overall picture these studies paint about the safety and effects of this specific PMA-Zeolite material in humans?
Well, I think the most prominent takeaway is the consistent safety profile observed across different populations and durations. The initial concerns about the material leaching significant amounts of contaminants like aluminum or lead into the bloodstream upon intake were not supported by the data, particularly the immediate post-dose testing.
Right, that 1-hour test was pretty clear.
And the concern about widespread, clinically relevant depletion of essential minerals also didn't materialize in the healthy volunteers or the Crohn's patients over short to medium terms. Their levels generally stayed within normal ranges.
So the core safety questions seem reasonably well addressed for this material.
Based on this evidence, yes. And alongside that safety profile, the studies collectively demonstrated a clear potential for detoxification of specific metals. We saw significant decreases in mercury and cadmium in the short-term MMBP study.
Arsenic in the medium-term Crohn's study.
And then robust long-term evidence for aluminum and nickel detoxification in the TOT-A study, plus that intriguing pattern suggesting lead mobilization and subsequent elimination over several years.
So it definitely seems capable of binding and helping remove a range of undesirable metals from the body. But the complexity revealed in the osteoporosis study regarding essential minerals is also important to remember.
Absolutely. It highlights that context matters. While generally safe regarding minerals in healthy people, the long-term use in a specific patient population, those with osteoporosis undergoing treatment potentially affecting bone metabolism, was associated with decreases below the normal range for copper, sodium, and calcium.
Potentially linked to the disease process or the treatment effect itself, as they hypothesize.
Right. And this led to the sensible recommendation for monitoring these specific minerals in that particular patient group during long-term use. It shows that while the material itself seems safe, its interaction with the body can be influenced by underlying health conditions and other treatments.
So the clinical picture from Source 1 is generally safe, demonstrated detox for specific metals, Hg, Cd, As, Al, Ni, and likely Pb over time, but with the nuance that long-term effects on essential minerals might need monitoring depending on the individual's health status, particularly in conditions affecting mineral balance like osteoporosis.
I think that's a fair and accurate summary of the clinical trial findings presented in Source 1.
Okay, now let's broaden our view a bit. Let's connect these clinical findings to some of the other potential health effects and mechanisms that are discussed in Sources 2 and 4, because it seems like this stuff might be doing more than just swapping ions in the gut.
Definitely. Sources 2 and 4 delve into several other interesting areas. A really major focus is its potential impact on gut health and the gut microbiota. It seems to interact quite significantly with the environment inside our intestines.
How does it do that, according to these sources?
Well, for one, it can absorb water, which might relate to its traditional use for things like diarrhea. More significantly, perhaps, it has a high affinity for waste products generated in the gut, like ammonia. Source 4 specifically highlights its ability to bind ammonium. It can also help remove other uremic toxins like urea and creatinine.
So cleaning up metabolic waste products right there in the gut.
That seems to be part of it. These substances can be irritating to the gut lining and potentially cause problems if they're absorbed systemically. And beyond just cleaning up waste, there's evidence it directly influences the bacteria living in the gut.
Ah, the microbiome connection. What does it do to the bacteria?
Sources 2 and 4 suggest it has a beneficial effect on the microbial population. They point to animal studies where giving animals Clinoptilolite led to a decrease in potentially harmful bacteria, like certain types of E. coli, and at the same time, an increase in beneficial bacteria, like lactobacillus and bifidobacterium species.
So shifting the balance towards the good guys, is there any human data that supports that?
Source 4 does mention findings from a human study involving patients with irritable bowel syndrome, or IBS. In that study, while the changes in specific bacteria weren't always large enough to be statistically significant across the group.
There were trends.
Yes, the group receiving PMA-ZC showed trends towards increased levels of beneficial bifidobacteria and lactobacillus, and also a reduction in the overall load of proteobacteria and enterobacteriaceae, groups of bacteria, often linked to gut inflammation and dysbiosis.
Okay, so hints in humans, too, that it helps foster a healthier microbial community. What about other effects on the gut itself, the structure or function?
Source 4 discusses potential improvements in the gut lining or morphology. Animal studies, for example, have shown things like increased height of the villi, those finger-like projections that absorb nutrients, suggesting better nutrient absorption capacity. It might also help lower the pH in parts of the gut, making the environment more acidic.
And why is lower pH good?
A slightly more acidic environment is generally more favorable for the growth of beneficial bacteria and for the optimal function of digestive enzymes, while being less hospitable for many pathogenic bacteria.
Okay, so healthier gut lining, maybe better nutrient absorption, better environment for good bacteria. It sounds like it generally supports a healthier gut ecosystem. The sources even mentioned something about antibiotics.
Yeah, that was an interesting point in sources 2 and 4. They proposed that because of its positive effects on gut bacteria and overall gut health, Clinoptilolite could potentially be explored as an alternative strategy or maybe a supportive one in places like animal farming. The idea is to reduce the need for antibiotics.
And maybe even impact antibiotic resistance.
Source 4 suggests it might even help reduce the abundance of antibiotic resistance genes within the gut microbiome, which is obviously a huge public health concern. They also mention another gut-level detoxification mechanism, its ability to absorb mycotoxins.
Lycotoxins, like mold toxin.
Exactly. Toxic compounds produced by certain molds that can contaminate food, like aflatoxins. Zeolite seems capable of binding these in the gut as well, preventing their absorption.
Wow. Okay, so it's doing a lot in the gut, improving the environment, balancing bacteria, helping remove waste products, ammonia, maybe mycotoxins. Now, this is where sources, particularly Source 4, make a really critical leap, connecting this gut health piece to something much bigger.
Yes, this is the connection to gut barrier function, or what's often called leaky gut. Source 4 emphasizes this strongly. We know that increased intestinal permeability is linked to gut problems like inflammatory bowel disease, which was relevant in the Crohn's study we talked about.
Right. But the link goes beyond the gut itself.
Absolutely. Source 4 states very clearly that a growing body of research is now establishing a significant link between this increased intestinal permeability, this leaky gut, and major neurodegenerative diseases like Alzheimer's and Parkinson's.
Wait, leaky gut linked to Alzheimer's and Parkinson's?
Yeah. The hypothesis, gaining traction, is that when the gut barrier becomes compromised or leaky, it allows unwanted substances to pass from the gut lumen into the bloodstream. Things like bacterial fragments, like LPS, lipopolysaccharide, which is highly inflammatory, undigested food particles, or inflammatory molecules.
And these things travel through the blood to the brain.
Potentially, yes. The idea is that these substances circulating systemically can trigger widespread inflammation, and some may even cross the blood-brain barrier, directly contributing to neuroinflammation and the pathological processes seen in diseases like AD and PD. Source 4 states this link between leaky gut and ADPD has been observed in patients.
That's a huge connection. A problem in the gut potentially fueling a problem in the brain. And Source 4 suggests zeolite might help with this leaky gut issue.
It presents evidence suggesting it can. They discuss findings from animal studies using chickens as a model, where clinoptilolite supplementation led to decreased levels of markers associated with gut permeability, things like circulating LPS, D-lactic acid, and an enzyme called DAO.
So less bad stuff getting through. Did it affect the barrier itself?
Yes, and this is really key. Those same animal studies showed increased expression of crucial tight junction proteins. These are the proteins that literally form the seals or glue between the cells lining the gut wall proteins, with names like ZO1, occludin, and claudin.
So it might be helping to physically strengthen the seals in the gut lining, making it less leaky.
That's exactly what those findings imply, decreasing markers of leakiness and increasing the structural components of the barrier. And Source 4 also points to human studies.
The athlete and IBS studies again.
Right, where zeolite supplementation was associated with decreases in markers, sometimes used to assess gut permeability and inflammation, like zonulin, alpha-1 antitrypsin, and that general inflammation marker, HSCRP.
Okay, so animal data showing structural improvements and reduced leakiness, and human data showing reductions in markers associated with permeability and inflammation. That sounds like strong support for improving gut barrier function. Are the exact mechanisms understood? Like, how does it do this?
Source 4 is careful to say that the detailed mechanisms aren't fully worked out yet. It's likely not just one single thing. They suggest it's probably a combination of zeolite's beneficial effects on the gut microbiota composition, its ability to bind and remove inflammatory triggers like ammonia or LPS within the gut, plus its own inherent anti-inflammatory and antioxidant properties, all working together to create a healthier gut environment that promotes a stronger, less permeable barrier.
Yeah, makes sense. It's probably a multi-pronged effect.
Yeah.
Let's dive a bit deeper into those other properties you mentioned. Antioxidant effects, for example.
Yeah.
Oxidative stress comes up a lot in chronic diseases.
It really does. Sources 2 and 4 both emphasize the role of oxidative stress, that imbalance between damaging reactive oxygen species, ROS, and the body's antioxidant defenses in the development of many conditions, including neurodegenerative diseases. ROS can damage proteins, lipids, DNA.
And zeolite might help fight this.
The sources present evidence that it can. In vitro studies, so in the lab, show that zeolites can act as direct ROS scavengers, meaning they can neutralize these harmful molecules. Source 4 specifically mentions PMA-ZC reducing ROS generated by mitochondria in a neuroblastoma cell line, which is relevant to brain cell damage.
Okay, direct scavenging in a dish. Does it work inside a living organism too?
Yes, there's evidence for that as well. Animal studies feeding Clinoptilolite have shown increased activity of the body's own major antioxidant enzymes, catalase, CAT, superoxide dismutase, SOD, glutathione peroxidase, GPX. These are the frontline defenders.
So it boosts our internal defenses.
It seems so. And consistently, those studies also showed decreased markers of oxidative damage, like less lipid peroxidation, damage to fats and cell membranes. Source 4 also mentions similar findings popping up in human trials. Supplemented individuals showing increased antioxidant enzyme activity and reduced markers of oxidative damage, particularly noted in studies involving smokers or athletes under stress.
So it might be acting as an antioxidant itself and boosting our own systems.
Right.
Any idea how it might boost our enzymes?
Sources 2 and 4 touch on a hypothesis. It suggested that the metal ions naturally present within the zeolite structure, remember those cations like calcium, magnesium, etc., might potentially act as cofactors, essentially helping to activate or enhance the function of these antioxidant enzymes, many of which require metal ions to work properly.
Ah, that's a clever potential mechanism.
Yeah.
Okay, antioxidant effects seem well supported. What about inflammation, another big player in chronic disease?
Absolutely. Chronic inflammation is a common thread in many conditions, and Source 4 explicitly links it to neurodegenerative diseases as well. And yes, zeolite appears to have anti-inflammatory properties too.
How does it seem to reduce inflammation?
Source 4 cites findings from cell culture and animal models where zeolite treatment counteracted key inflammatory pathways. It reduced levels of pro-inflammatory signaling molecules called cytokines, like TNF-alpha and IL-1 beta, and it inhibited the activation of NF-kappa, which is like a master switch for turning on inflammatory genes.
So interfering with the inflammatory signals.
Right. And Source 4 also mentions an interesting finding where zeolite seemed to enhance the effect of a known anti-inflammatory drug, diclofenac, suggesting it might work synergistically.
Is there human evidence for anti-inflammatory effects?
Going back to those human studies in athletes and IBS patients mentioned in Source 4, they reported associated changes in inflammatory markers. Things like a trend towards increased levels of IL-10, which is an anti-inflammatory cytokine, and decreased levels of hsCRP, that general marker of systemic inflammation we mentioned earlier.
Okay, so add antioxidant and anti-inflammatory actions to the list. What about the immune system more broadly? Did the sources mention effects there?
Yes, Sources 2 and 4 also discuss the potential for zeolites to modulate or influence the immune system, particularly through interactions with the gut-associated lymphoid tissue, the GELT-T.
The immune system in the gut lining.
Exactly. It's a huge part of our overall immune system, constantly monitoring what's going on in the gut. Animal studies, mentioned in Source 4, have shown that Clinoptilolite supplementation can lead to increases in certain types of immune cells, like B lymphocytes, which make antibodies, and increased levels of specific antibodies important for gut defense, like IgA and IgM.
So potentially strengthening the immune defenses right at the gut barrier.
That seems to be the implication. And there's some human data, too. Sources 2 and 4 refer to a study in immunodeficient patients where taking tribomechanically micronized clinoptilolite was associated with increases in the counts of certain immune cells, including B cells, T helper cells, and activated T cells.
That sounds like it could be broadly supportive for immune function. Any ideas on how it might be doing this?
Source 2 and 4 mention a couple of possibilities. One is that the zeolite particles might interact with specialized cells in the gut lining called M cells. These M cells actively sample particles from the gut lumen and present them to the underlying immune cells, initiating responses.
So maybe the zeolite particles trigger a beneficial immune sampling.
That's one idea. Another, more speculative mechanism mentioned in the sources is the possibility that zeolite could act as something called a silicate superantigen. Superantigens are substances, often from bacteria, that can cause a very strong, nonspecific activation of immune cells. The idea here is that the silicate structure might trigger a similar, perhaps beneficial, immune stimulation.
Wow. Okay, let's recap the mechanisms beyond simple ion exchange. We've got improving the gut environment, balancing the microbiota, reducing harmful waste products and maybe mycotoxins, strengthening the gut barrier, potentially reducing leaky gut, acting as an antioxidant, reducing inflammation, and possibly modulating immune function, especially in the gut. That's a lot for one mineral.
It really paints a picture, based on these sources, of a material with quite multifaceted biological activities.
Now let's bring it all together with that critical connection we touched on earlier. The link between heavy metals and neurodegenerative diseases. This is where things get really compelling, I think. Source 4, in particular, makes a very strong case linking environmental factors, especially metals, to diseases like Alzheimer's and Parkinson's.
It absolutely does. Source 4 lays it out quite clearly. Neurodegenerative diseases, and they list AD, PD, Huntington's, ALS, MS, are understood to arise from a complex mix of our genes and our environment. And within those environmental factors, pollution and specifically exposure to metals are highlighted as really significant contributors.
And Source 4 gets specific about which metals are most implicated. Let's start with the obvious bad guys. Lead and cadmium.
Right. Lead, Pb, and cadmium, Cd, are described flat out as neurotoxins directly harmful to nerve cells. Their toxicity involves multiple hits, they cause oxidative stress, they trigger inflammation, and they mess with mitochondria, the energy factories in our cells. All of this can lead to neuron damage and death.
And specific links to ADPD.
Source 4 mentions cadmium specifically promoting the clumping, the aggregation of amyloid beta, which is that key protein in Alzheimer's plaques. And for evidence, it cites animal studies showing early lead exposure leads to AD-like brain changes later on, including more AVI and tau protein and cognitive problems. In humans, studies have linked higher lead exposure to cognitive decline and cadmium exposure to lower cognitive function and higher AD risk. Source 4 even mentions acute cadmium poisoning causing Parkinson's-like symptoms.
Okay, so lead and cadmium are clearly bad news for the brain. What about those metals that we actually need in small amounts but can be toxic if we get too much, like iron, copper, manganese, zinc?
That's a crucial category. Iron, Fe, copper, manganese, Mn, and zinc, Zn, are essential micronutrients, vital for countless processes at the right levels. But Source 4 really emphasizes that when the body's handling of these metals goes wrong and they accumulate in the wrong places or amounts, they become toxic and contribute significantly to neurodegenerative processes.
How do they cause damage when they're in excess?
Similar mechanisms to lead and cadmium, actually. They strongly promote oxidative stress. They can cause essential proteins to misfold and clump together, like AF and alpha-synuclein, and they fuel inflammation in the brain.
Let's take iron. We need it for blood, but too much in the brain.
Can be a problem. Source 4 notes that increased iron levels have consistently been found in the specific brain areas damaged in both Alzheimer's and Parkinson's patients. Iron is known to directly promote the aggregation of AF in AD and alpha-synuclein, the key protein in PD pathology.
And manganese. I know high exposure causes problems.
Yes. Excessive manganese exposure causes a condition called manganism, which presents very similarly to Parkinson's disease. Source 4 also connects manganese to AD pathology, AT and tau changes, and cognitive decline in some studies, and mentions potential links to ALS and Huntington's as well.
Okay, now copper and zinc. Source 4 seemed to really emphasize their role, particularly copper and Alzheimer's.
It did. Source 4 states that problems with the body's regulation, the homeostasis of copper, C, and zinc, Zn, are pivotal in both AD and PD. While zinc levels are often found to be lower or dysregulated in AD, Source 4 highlights that copper excess is the metal most consistently linked to Alzheimer's disease in the reviewed research.
Copper excess specifically, why is it so bad for AD?
According to Source 4, Copper actually accumulates within the amyloid plaques and the neurofibrillary tangles made of tau protein in AD brains. It seems to actively promote the formation of AP plaques and the development of tau tangles. Copper binds directly to the apopeptide, and when it does, it can trigger chemical reactions that produce highly damaging reactive oxygen species, contributing massively to the oxidative stress seen in AD.
And in Parkinson's.
In PD, excess copper is also directly toxic to dopamine-producing neurons and promotes the aggregation of alpha-synuclein. Source 4 also notes that often in AD patients you see a pattern of lower zinc levels alongside higher copper levels.
Wow. The balance of these essential metals seems incredibly delicate when it comes to brain health. Now, what about aluminum? Source 4 treats it as a major neurotoxin, even though it's not technically a heavy metal.
Right. Source 4 makes a strong statement. Aluminum, Al, is a known neurotoxin with no known beneficial biological function in the human body. And the research linking aluminum exposure to Alzheimer's goes way back to the 1960s. Source 4 reviews considerable evidence supporting this connection.
What does aluminum actually do in the brain that's so bad?
It interferes with a lot of critical functions. Source 4 mentions it disrupting the neurotransmitter acetylcholine, vital for memory, messing with calcium signaling in neurons. Animal studies clearly show aluminum exposure causes memory and learning problems. Crucially, aluminum accumulates in the plaques and tangles in AD brains.
It gets right into the pathological structures.
Yes. And Source 4 emphasizes that aluminum doesn't just inhibit the enzymes that normally break down A, it also enhances the toxic aggregation of A, potentially more effectively than other metals like zinc, iron, or copper. It also speeds up the development of tau pathology. And beyond AD, aluminum exposure has also been linked to ALS and multiple sclerosis.
Okay, so Source 4 paints a really convincing picture. Lead, cadmium, excess iron, manganese, copper, zinc dysregulation, and aluminum are all strongly implicated through various mechanisms like oxidative stress, inflammation, and protein aggregation in the development or progression of neurodegenerative diseases.
That is the very clear connection established in Source 4 based on a review of the scientific literature.
And this is where we circle right back to zeolite clinoptilolite, because its demonstrated ability to bind and remove several of these specific metals suddenly seems incredibly relevant, doesn't it?
It really does. Sources 2 and 4 explicitly state that zeolite has a high affinity for and can help remove many of these implicated metals. Lead, cadmium, arsenic, aluminum, nickel, copper, zinc, iron, cobalt are all mentioned as being bound by clinoptilolite.
And the clinical trials from Source 1 that we just discussed provided actual human evidence for the removal of some of these key players.
Exactly. We saw a demonstrated detoxification of aluminum and nickel confirmed in the long-term TRP study. We saw a mercury and cadmium decrease in the short-term MMBP study. Arsenic decreased in the medium-term Crohn's study, and that complex pattern for lead in the TOTAP study strongly suggests mobilization and eventual elimination.
So while nobody is calling it a cure, the ability of this specific PMA-Zeolite to help lighten the body's load of these potentially neurotoxic metals, it suggests a plausible supportive role, doesn't it, by potentially addressing one of the environmental risk factors these sources highlight.
That's the really fascinating implication when you connect the dots across these different sources. By assisting the body in eliminating some of these metals linked to neurodegeneration, it might help mitigate one piece of that very complex puzzle.
Okay, so that's the indirect link, reducing the burden of toxic metals.
Mm-hmm.
But did the sources, particularly Source 4, provide any direct evidence suggesting zeolite could impact the actual disease processes in the brain, beyond just metal removal?
Yes. Source 4 does present some very interesting, though preliminary, findings that hint at more direct effects. It's important to stress, though, that these are mostly from preclinical studies, lab experiments, or animal models, not yet proven in human patients with these diseases.
Understood. Preclinical, but still potentially informative. Tell us about that study mentioned in Source 4 using an Alzheimer's mouse model.
Okay, this study used a specific type of mouse, the APP-SWEP-S1DE9 model. These mice are genetically engineered to develop amyloid plaques in their brains as they get older, mimicking a key feature of human Alzheimer's.
Right. What do they do?
They gave these mice PMA-Zeolite dissolved in their drinking water for a period of 5 months, and the results reported in Source 4 were pretty remarkable.
What do they find?
Compared to the control mice that didn't get the zeolite, the treated mice showed a significant reduction. Source 4 says over 50% in the levels of the toxic A42 peptide, in the actual number of amyloid plaques counted in the brain tissue, and in the overall amyloid load or burden.
Wow. A 50% reduction in amyloid pathology in an Alzheimer's mouse model. That seems huge, even if it is preclinical.
It's certainly a striking finding. And intriguingly, the researchers linked this reduction in amyloid pathology directly back to the zeolite's antioxidant activity.
How do they make that link?
In the same brain regions where they saw less amyloid, they also measured increased activity of antioxidant enzymes, like SOD, and they saw reduced markers of oxidative damage in the zeolite-treated mice. This tied in with their lab dish findings showing PMA-ZC reduced mitochondrial ROS production.
So the thinking is, by reducing oxidative stress in the brain, the zeolite might be directly interfering with the processes that lead to amyloid buildup, or maybe protecting neurons.
That's the suggested mechanism presented in Source 4. And there was another relevant animal study discussed too, using a different model.
Right, the one using aluminum chloride to induce AD-like changes in rats.
Exactly. In this model, exposing rats to aluminum chloride causes cognitive problems and biochemical changes in the brain that resemble Alzheimer's. The study found that giving these rats zeolite supplementation alongside the aluminum exposure significantly counteracted many of the negative effects of the aluminum.
Counteracted how? What specifically did the zeolite improve?
It helped normalize several key markers. It decreased harmful things that were elevated by the aluminum, like the activity of acetylcholinesterase, an enzyme that breaks down a memory-related neurotransmitter, levels of N-peptide itself, inflammatory markers like TNF-alpha and NF-kappa activation, and signs of cellular damage from oxidative stress, lipid peroxidation.
So it reduced the bad stuff. Did it boost any good stuff?
Yes. It also increased beneficial markers that the aluminum had depleted. Things like brain-derived neurotrophic factor, or BDNF, which is crucial for keeping neurons healthy and supporting learning and memory, and another marker called phosphorylated GSK3 beta.
And did these biochemical improvements translate into better brain function for the rats?
They did. The source reports that these positive biochemical changes were accompanied by significantly improved performance in learning and memory tasks, like the Morris Water Maze test, for the rats receiving zeolite compared to those getting only aluminum.
That's really compelling evidence from an animal model suggesting it might protect against aluminum's neurotoxic effects and the resulting cognitive decline. Were there any lab dish studies looking at AD pathology?
Source 4 also mentions some in vitro work experiments done in test tubes or cell cultures looking at how zeolite affects AT aggregation directly. One interesting finding was that the effect seemed to depend on which metal cation was loaded into the zeolite structure. Specifically, zeolites containing copper, CaOy, or zinc, ZNY, were shown to inhibit AT aggregation.
Inhibit clumping.
They seem to stop the process from forming the mature fibrillar amyloid plaques, arresting it at an earlier stage involving smaller clumps called oligomers. Some research suggests these oligomers might actually be the most toxic forms of ag, so interfering with that process could be beneficial.
That is interesting, especially given the complex role of copper and zinc in AD we just discussed. So the zeolite itself, depending on its composition, might directly interfere with the protein misfolding. Any other in vitro findings?
Yes. Source 4 also notes that zeolite nanoparticles were shown to inhibit the interaction between apoptide and fibrinogen in lab tests. Fibrinogen is a blood-clotting protein.
Why is that interaction relevant?
This interaction is thought to contribute to the formation of abnormal blood clots and microvascular problems often seen in the brains of Alzheimer's patients, which can worsen cognitive impairment. So blocking that interaction could be another potential protective mechanism.
Okay, so these preclinical findings, specifically related to Alzheimer's models, reducing amyloid plaques in mice, protecting against aluminum toxicity and cognitive decline in rats, potentially interfering with AT aggregation and fibrinogen interactions in vitro, they are definitely intriguing, even with the caveat that they are preclinical. Now, what about Parkinson's disease? Did the sources present any direct evidence of zeolite impacting PD pathology, like the alpha-synuclein protein, in models?
The sources we have here, particularly Source 4, which discusses the ND links, do not present direct studies showing zeolite treating the core pathology of Parkinson's, like reducing alpha-synuclein aggregation or protecting dopamine neurons in animal models, in the same way they showed for Alzheimer's models.
So no direct treatment evidence for PD models.
Not presented in these specific sources. However, Source 4 does mention a couple of potential related applications involving zeolites in Parkinson's.
Such as?
One area is in developing better diagnostic tools. Zeolites have apparently been used experimentally in constructing specialized electrodes for biosensors designed to detect LDOPA and dopamine, the key molecules involved in PD.
Okay, diagnostics. Anything else?
Source 4 also mentions a more speculative idea and stresses there's no clinical data supporting it yet of potentially using zeolite as a kind of carrier material. The idea would be to load it with the drug L-DOPI to try and help transport it more effectively across the blood-brain barrier into the brain where it's needed.
So interesting ideas for potential applications in PD diagnostics or maybe drug delivery, but no direct evidence presented here yet that zeolite itself modifies the underlying disease process in PD models like it seemed to in the AD model.
That's correct. Based purely on the information within these provided sources, the direct preclinical evidence concerning neurodegenerative diseases focuses primarily on Alzheimer's models and mechanisms.
Okay, well, we have certainly covered a huge amount of ground here. We started with the basic structure and ion exchange properties of clinoptilolite. We went through the detailed findings of 3 human clinical trials, looking at the safety, mineral balance, and detoxification effects of a specific PMA-ZC. We explored the broader potential health mechanisms suggested by reviews improving gut health, strengthening the gut barrier, antioxidant and anti-inflammatory effects, maybe immune modulation. And then we tied it all together by looking at the critical role heavy metals and aluminum appear to play in neurodegenerative diseases, and finally examined that intriguing, though preliminary, direct evidence suggesting zeolite might impact AD pathology in preclinical models.
It is indeed a complex picture, weaving together chemistry, clinical science, gut physiology, immunology, and neurobiology. And as we wrap up, it's really vital to circle back and remember the context and the limitations that the sources themselves were careful to point out.
Absolutely. Source 1, for instance, was very transparent about the limitations of the clinical trials. The MMBP and the Morbus Crohn studies, being smaller pilot or exploratory studies, had sample sizes that limited their statistical power to draw really definitive conclusions about subtle changes in mineral balance for everyone.
Right. And factors like the participants' different dietary habits, Mediterranean versus continental diets, seem to influence some of the baseline metal levels, like mercury or arsenic, suggesting that results could potentially vary depending on a population's background, exposures, and lifestyle.
And that crucial point, emphasized in both Source 1 and Source 4, is that all these clinical findings apply specifically to one particular type of processed material, the PMA-ZC.
Yes, that can't be overstated. Because this material undergoes that specific patented tribomechanical activation process, you simply cannot assume that the safety profile, the effects on minerals, or the detoxification results observed here would be identical for other forms of natural clinoptilolite that are unprocessed or processed using different methods.
You need separate research on those other products.
Exactly. Additional dedicated studies would be required to confirm the properties and effects of any other specific zeolite material.
So while the findings are promising, they are specific to this well-defined, prepared, and tested material. Given all this, what did the sources conclude about the next steps? What future research is needed, especially regarding these potential links to neurodegenerative conditions?
Both Source 1 and Source 4 conclude on a similar note. While the tested PMA-ZC material appears generally safe based on these trials and shows some really intriguing effects, detoxification, gut health support, antioxidant activity, they both stress that more research is definitely warranted.
And is that future research specifically targeting neurodegenerative diseases?
Yes, Source 4, in its conclusion, explicitly calls for the need for dedicated studies in relevant animal models of specific neurodegenerative diseases, suggesting, for example, more extensive work in AD models and initiating studies in PD models. And even more critically, Source 4 highlights the need for well-controlled clinical trials conducted in human patients actually diagnosed with these conditions, like Alzheimer's or Parkinson's.
Because the trials we looked at today weren't designed for that.
Precisely. The clinical trials presented in Source 1 were primarily focused on establishing general safety, understanding the effects on mineral and metal levels in specific groups, healthy osteoporosis, Crohn's, and exploring detoxification potential. While they provided invaluable human data on those fronts, and the long-term TOP study gave safety reassurance over years, they were not designed to assess whether PMA-ZC could alter the course, symptoms, or underlying pathology of neurodegenerative diseases in affected patients. That's a different and much more complex research question that needs its own dedicated trials.
Okay, so putting it all together from these sources, we have a natural mineral material, specifically PMA-ZC, which has demonstrated safety in human trials. It shows clear ability to bind and help remove certain heavy metals and aluminum. It seems to positively influence factors strongly associated with neurodegenerative risk, things like gut barrier integrity, inflammation, oxidative stress. And there's some exciting, though still early, preclinical evidence hinting it might even directly impact AD pathology in models. But the crucial next step, the bridge to confirming any real potential for treating or preventing these diseases in humans, requires those dedicated clinical trials in patient populations.
That is a very accurate and nuanced synthesis of the state of the research as presented within these specific sources we've examined today.
Well, we started this deep dive talking about that feeling of being overwhelmed by information, trying to figure out what matters. We took your stack of sources on zeolite, clinoptilolite, and hopefully, by spending this time navigating through them together, we've pulled out the key insights and put them into perspective.
I think we've seen that these particular studies and reviews really do paint a picture of a natural material with some demonstrated safety in humans and some potentially significant biological effects. That ability to detoxify certain metals seems quite robust based on this evidence. And the influence on gut health, particularly strengthening that crucial gut barrier, is supported by multiple lines of evidence from animals and humans in these sources.
And we've made those connections, as laid out in the sources, to the way we're increasingly understanding complex brain diseases like Alzheimer's and Parkinson's, where factors like the burden of heavy metals, the health of the gut, chronic inflammation, and oxidative stress are seen as playing these deeply interconnected roles.
That preliminary direct evidence from the 80 animal models showing actual reduction in amyloid pathology and protection against aluminum toxicity is certainly compelling. It definitely suggests the potential for this material to play a supportive role, maybe by addressing some of these underlying factors, or perhaps, as those animal studies hint, even influencing the pathology itself.
But, and it's a crucial but that the sources themselves make clear, the science is still evolving. More dedicated, high-quality research, especially those controlled clinical trials, focusing specifically on outcomes in patients with neurodegenerative conditions, is absolutely essential to truly understand if and how this material might fit into future health strategies for tackling these devastating diseases.
So what does this all mean for you listening in? It means the research around zeolite clinoptilolite, particularly this activated form, is opening up some really fascinating questions. It pushes us to think more about that intricate dance between our environment, our gut health, inflammation throughout our body, and ultimately the health of our brain. It makes us consider how relatively simple natural materials with their multifaceted actions could potentially contribute to supporting overall well-being, especially as we face rising rates of complex chronic diseases.
And maybe here's a final thought for you to take away, something to mull over based on the connections we've explored today from these sources. Given this incredibly complex web of factors, genetic, environmental, lifestyle, that we now know contribute to neurodegenerative diseases, how much of a difference could interventions targeting fundamental processes actually make? Things like reducing our exposure to or burden of environmental toxins like heavy metals, or actively supporting the health and integrity of our gut barrier. Are these potentially overlooked avenues for support while the search continues for highly specific, single-target cures? Is there value in exploring these broader, potentially foundational strategies found in nature?
Thank you for joining us on this deep dive into the research on zeolite clinoptilolite.
Keep asking those questions. Keep exploring the sources.